Before I start with, I'd just like to introduce all our participants about Venture Center, who we are, what we do, and from there, then I'll introduce about our service offerings, and then I'll speak about more about our speaker and the title for today's session. Venture Center is India's leading technology business incubator. We are a Section 8 not-for-profit organization, and we are hosted by CSR NCL, National Chemical Laboratory, and we are approved by NSTDB and BIRAC, both Government of India entities. So, as a technology business incubator, we aim to provide a complete ecosystem for deep tech and science-based startups. That said, although our primary support system is meant for startups, we operate as an open access facility. That means that anybody who needs any of our services, we are open to assist and helping them with their requirements. So, when I say complete ecosystem, what exactly do we provide? So we offer mentoring support to the startups and any innovators who need to build or further develop their products. We connect them to various funding agencies and in the forms of fellowships, POC, and prototyping grants. Since these are science and tech-based startups, we offer infrastructural facilities such as dedicated labs, office spaces, and event spaces. So they can come get in their own equipment and do their initial R&D in these empty shells that we provide. We also have a good collection of various funding databases, and we have partnerships with various other facilities like design and prototyping, trial manufacturing, finance, and legal. So all this is required at the initial stage of the startups in their entrepreneurial startup journey. Now, coming to the heart of the incubator is the scientific facilities and the scientific services that we offer. So we have plug and play labs, which where we house 100 plus workhorse equipment, where people can just come and use it at a very nominal rental that is available. So initially, the startups don't have to invest in it, but they can make use of these equipments to build all their prototypes or build their processes or build their molecules. The details of all these equipments are on the website. I would urge all of you all to kindly visit and have a look at all the details there. Then we have a well-established prototyping facility where we have all the equipments like 3D printers, then the laser scanners, CNC machines. So all of this, since some of our startups are working in the medtech domain, if they want to build their prototypes, all the facilities that are required are available there. Then we have our testing and analysis is horizontal, where we have certain core analytical equipment, and we have the advanced and certified resource centers, where we offer all the analytical based service offerings, details of which I'll be explaining in the subsequent slides. Then we have the regulatory facilitation center, which provides the mentoring and support to navigate through the regulatory requirements of each of our startups. We have a tech transfer and IP facilitation center as well, and we are aligned with many national programs that help our startups at various juncture at in their journey. Coming to the analytical services, so as I mentioned, we have certain core equipments like GCs, HPLCs, elemental analysis, and these we offer as a full service to anybody who requires it. So yes, startups are one domain that we serve, but in addition to that, any researchers or academic institutes and even major industry players all are availing of our services. We operate as an open access facility, and there are special discounts for startups and students. All the detailed list of our machines, their make, vendor types, and our pricing is on the website. So if any information is required, you can have a look on the website. Coming to our advanced and certified facility at Venture Center, so is our Center for Biopharma Analysis, which is a GLP compliant and NABL accredited facility, one of its kind in the entire country. So the CBA, as we call it, is a one-stop destination for the characterization of biopharmaceuticals. Again, CBA is an open access facility, so any startups or even big players like big multinational players working in the biopharma domain can avail avail and have been availing our services because we follow OECD GLP guidelines and also ISO 17025 guidelines as well. Then we have all the high-end equipment under one roof. So any of right from the primary order to the higher structure order, all the analysis can be carried out at our facility. In addition to this, we also provide the hand-holding and advisory support. So, we have something on a scientific consultation. So in case people need some, assistance with the data analysis or how to, design experiments, all that we are provided. And we are supported by the National Biopharma Mission of the Government of India. So that was about CBA. As I mentioned, at CBA, we can do both the structural analysis and the functional analysis. So right from the primary order to biosimilarity assessment, we have worked on around 20 projects and characterized almost 15 plus molecules till date. And on the functional side, we have a whole range of assays that we have not only developed but also validated with really high quality data coming out. So these are already readily available for the service offerings. This is just a list of all the equipment that are at the CBA facility. So we have an HRMS system, we have FTIR, we have an SPR system, capillary electrophore system, fluorescence spectrometer. Then we have the SEC-RI MALS, and the talk today is around this particular technique and equipment. In addition, we have UHPLC, CD spectrometers, nano DSC, plate readers, and stability chambers. Just wanted to mention, since this is a GLP compliant facility, all the equipment are 21 CFR compliant and they are all qualified, and we have a dedicated quality assurance unit that takes care of all the compliance. We have a Waters, SEC system with UV, RI, and MALS detectors attached to it. Definitely, we all know that absolute determination of molar mass is, possible, and characterization of various range of biomolecules can be done. Our speaker for today, Dr. Leo, is going to cover all this in detail, so I'll keep it very brief. But this machine is recently installed at Venture Center, as it has been upgraded with the RI detector, and we have successfully delivered on the commercial projects as well. So with that brief introduction of Venture Center and our scientific facilities, I'll come to our speaker for today, and we are very happy to have with us Dr. Leo, who is a senior scientist at Waters Corporation. Thank you, Dr. Leo, so much for accepting our invitation to deliver this session today. So Dr. Leo obtained his PhD from UCLA in biochemistry, where he and his research group designed synthetic enzyme systems capable of carbon fixation and production of useful biomaterials. During his PhD, he regularly utilized SEC-MALS for quality control of enzymes and other protein elements. After his PhD, he co-founded a startup company which was focused on making LNP as food additive backed by venture capitals and the Hult Prize Foundation. Now at Viat Light Scattering Portfolio for Waters APAC, he is a resident expert. He resides in Singapore while he loves to travel to India, Japan, Korea, and other Asian countries. His expertise is in biopharma and materials application with light scattering techniques. Besides his passion for biopharma applications in his private life, he loves skiing, scuba diving, and photography. So we can see he's a multifaceted personality with us today. So with that, I'll now take a pause. I'd also like to say that, we are available on social media platforms like LinkedIn, X. We have a WhatsApp group, and this session today will be recorded and uploaded on our YouTube channel, so you all can refer to it for future, references. And any of your queries, you can use this barcode to scan and reach out to us, and we'll be happy to assist you all ahead. So with that introduction, I'll now invite our speaker, Dr. Leo, to now take the session ahead. Dr. Leo, over to you.
Thank you very much. Thank you for the kind introduction. Now, I guess without further ado, I'll start my presentation.
Sure.
Now, do let me know if you are seeing the slides, and also, feel free to interrupt me during the entry. So thank you. My name is Leo. I'm a senior scientist at APAC Waters Corporation, and today I really wanted to go over some big ideas of how you can utilize light scattering along with SCC, that is Science Exclusion Chromatography. I'll touch a little bit on other mode of separation, such as reverse phase chromatography with light scattering or ion exchange chromatography with light scattering. But the main focus today will be with SCC. And how does that help with your biosimilars, such as proteins or peptides workflow? Briefly, a very, very high-level overview. Light scattering seek to understand these following parameters. Now, this is a very big table, but today we're going to focus mainly on here, here, and here. And I will skip these parts. So maybe in the future, in the series, we can do more here. So first, light scattering measures molar mass. And with molar mass, what we seek to understand is the identity of those species in its native conditions, such as whatever the formulations that you have, what are the different species, and how much there are. And light scattering is uniquely sensitive to aggregation, as in the larger the particles, the more light they scatter. So it's easy for us to see if there's any aggregates. And these aggregate, again, is in the context of native condition. With a little bit more careful planning of the experiment, such as looking at the size as a function of molar mass, right? Here you can see the size is increasing in radius, and the molar mass is also increased in grams per mole. And we are able to analyze the compactness or conformation of the molecules. Now, for protein, they generally behave globular. But should you ever work on, say, polysaccharide vaccines, this is where it becomes quite important. And lastly, since we are able to measure molar mass, and if we are to get concentration sources from two different species, for instance, like ADC, AOC, that's antibody drug conjugate, antibody oligonucleic acid conjugate, or like conjugate vaccines, like a polysaccharide protein conjugate vaccine, pneumococcal vaccine, BA1, or other like conjugate systems, then we are able to resolve molar masses from both parties. And from that molar mass, we naturally get the dosing information, such as the DAR, right, drug antibody ratio for ADCs, or like VGCP for AAV particles. Now, focusing on antibody, which most of my today's talk will be centered around it, and our favorite molecule. Again, the most important parameters are identity, aggregation or fragmentation, formulation stability, payload dosing if it's conjugate system. Today, I will not go into detail, just as a background information. Besides multi-angle light scattering with SEC, there are also plate-based approach, such as Nanostar, where samples are introduced on a cuvette, or plate reader, where sample is spotted on a microwell plate, or Zeta style, which also allow you to measure electrophoretic light scattering, that's Zeta potential, along with automation suite. So if you work in formulation development with lots of condition screening, this might be a better workflow. Such as stability, self-interactions, time course, freeze-thaw cycles, temperature ramp, these kind of things. Now, stepping away from the batch-based, and today, everyone here is to listen to size exclusion chromatography and the combination with MALS. So let's begin with that. As a quick refresher, I think everyone in the audience probably already know that light scattering, size exclusion chromatography fundamentally work based on RH, rate hydrodynamic radius. So large particles with a very large hydrodynamic radius are sterically excluded from entering or diffusing into the pores of the column packing material. So in green, this is the column packing material. So effectively, they travel the shortest distance. They just go through the column because they never diffuse in and out. Now, the smaller particles will then diffuse in and out of pores, making their travel distance longer, hence they elute later. So in SEC, you should expect larger things come out first and smaller things come out last. And then historically, age-old USP guidance, right? General Chapter 219 has been around for many, many decades. The idea is that if you're able to calibrate the column based on elution volume and the known molar mass, you should expect a certain molar mass calibration standard, a curve. However, not known at that time, and to this day, unfortunately, still a lot of people make these assumptions wrongly, is that there are 4 critical assumptions being made for this column calibration. Number 1, the calibrant, that's the standard, and the analyte, so intended target, has to have the same conformation. So imagine for a second, like the calibration is using a linear polymer, whereas the sample is highly branched polymer. They would have very different diffusion profile. Hence, their elution order or elution time will not reproduce the molar mass. 2, the standard and the sample must have the same density. So again, imagine, say, your analyte are ping pong balls, table tennis balls. That's our balls that's empty, very low density. However, the standard are bowling balls or something really hard with high density. Now, they might have the same hydrodynamic radius, right, of the same size. So they would have the same diffusion coefficient. However, because of density difference, their molar mass could be drastically different. And hence, the second assumption is same density. 3, we have to assume that there's no interaction with the column packing material. And 4, we have to also assume that there's no reversible interactions or self-interactions. Only when 4 critical assumptions are met simultaneously, where you have a perfect calibration. However, whether implicitly or explicitly known, these 4 assumptions are very hard and stringent to meet simultaneously. Hence, that's what we see. Very often, the elution volume and molar mass are irreproducible. Now, this is using a continuously polymer so that it draws a line. If it's a protein's globular, you'll just be dots. But basically, the point is that we have molar mass on the y-axis and elution volume on x-axis. As you can see, for a same elution volume, the molar masses sometimes could be 1 order of magnitude away from each other, right? And that's across different kinds of polymers. However, if you would consider the same polymer, polyethylene, this commercial polyethylene, which is in magenta, versus a standard polyethylene, NIST polyethylene, you can see even in that case, just both polyethylene, the elution order or the elution volume generated irreproducible molar masses. Again, a very substantial differences as well. So that is why we add light scattering after SCC, so that we rectify this irreproducibility and get true molar mass, regardless of elution. So the principle is that everything in this world is scatter light. So if we shine upon your sample with a laser, it will scatter light in all directions. The re-ligand theorem of light scattering dictates that the intensity of the light scattering is proportional to the concentration of the scatterer times the molar mass of the scatterer and this DNDC constant, which is refractive index increment. Now, this is a constant for known samples, such as protein, 0.185, polysaccharide, 0.135. These are well-known numbers. Concentration can be measured online, so that's why we have oftentimes a UV or RI detector. And light scattering, of course, is measured by the Dong multi-angle light scattering detector. So that leaves the only variable that's unknown here, and that's solved in the software that give you the molar mass. So measured, already know the last parameter here, M, comes out right away. And so for a protein analysis, this is how the system's typically set up. You would have a HPLC front end that provides the separation and also the UV detection units. For most proteins, they do absorb UV and the UV extension coefficient are known. And then you will have a light scattering detector plumbed right after. And then you have a RI detector, which is good for one, as a universal concentration detector. Two, it can be used in conjunction with UV to provide conjugate system analysis, such as a polysaccharide vaccine, right? Where the polysaccharide part does not absorb UV and the conjugated protein does absorb UV. So you have UV-RI simultaneously to work on the concentration. And that gives you molar mass, concentration, composition, if it's conjugate, aggregation impurities, extension coefficient, and size or conformation, all in a single assay. The data would look like this. So most of you, if you're familiar with chromatography, you will see a chromatogram like this, right? Now with MALS editing, you have a somewhat more complicated chromatography. You have a light scattering chromatography, and you have your concentration chromatography. So either UV or I. And once you have concentration and UV, concentration and light scattering, what do is that we analyze the molar mass, slice by slice, meaning that on the entire chromatogram, right? Typically 0.5 seconds, every 0.5 seconds, we make one measurement, and that gives you the molar mass. And then the next 0.5 seconds, we get another molar mass, so on and so forth. And so the beauty of that is that we are able to get a weight average molar mass of the entire peak, but also the nuances of distribution, whether it's homogeneous, heterogeneous, if there's co-elution, everything, because we are getting molar mass every 0.5 seconds, typically. And they are coming more with compendial support. So right now, if you go to the United States Pharmacopoeia, map analytical guide under release and stability and size variant, you will start to see secmoles. And if you go to the extended characterization, go to size variant, you will also see secmoles. Now, here is some real examples regarding to a simple map, and that's how we offered help to this University of Texas Austin group. One thing that we have immediately started to realize is, just like mentioned earlier, elution order is irreproducible. So here in red is bovine serum albumin, or BSA. And in blue is this research antibody. Now, conventional SEC wisdom would tell us the antibody is larger, it should elute earlier. But when we overlay the chromatography, we see that the antibody is eluting too late. Now, if we are relying on a traditional SEC, we'll never get to the end of it. Like, what is this peak, right? However, we can measure molar mass, right? As you can see this dot, as the chromatography elutes. And so we can see that indeed BSA is the monomer, here BSA is the dimer, because we know the molar mass now, trimer, and indeed this is the monomer antibody. So why it is eluting too late? Maybe this is due to column interaction. A lot of antibodies are quite sticky and they might interact with the hardware. But regardless, as they elute, we're able to measure its correct molar mass. Now, when we deal with a whole bunch of antibodies, so this is with a company, and this company is investigating a bunch of different biosimilars. And one challenge for them is that they want to have a platform method so that regardless of the elution time, so that they don't have to redevelop the chromatography, we're able to measure the molar mass and hence compare biosimilar to innovator antibodies. And so here is a big overlay of a whole bunch of different kind of antibodies. And as you can see, regardless of where they elute, we're able to accurately capture the molar mass and the nuances as well. Since the molar mass is measured every 0.5 seconds, we can see that for this blue antibody, this tailing in fact contains a fragment species, right? We can see the molecular weight right here, level of co-elution, and here is the fragment species, right? We can also see that for this brown antibody, right, where we have the monomer right here, and then we have a fragment that is fully baseline resolved. So again, this gives the customer the confidence in molar mass identity of the antibodies with a very minimal need for chromatography optimization, basically plug in and play. And this we've been working with the United States Pharmacopoeia. So for a lot of antibodies, we also see this kind of a peak asymmetry. And the big challenge is understanding what is causing it, right? Is it just column interaction, hardware interaction, or something quite fundamental to the sample that's going on? So what we have done is that we look at the molar mass of Remicade, and you can see this strange peak shape indeed also correlates with a very unique curvature of the molar mass trace. The molar mass is lower on the two ends and it's higher in the middle, colliding with the apex of the peak. Now, if we are to inject more and more and more, we can see it follows a very clear trend. As you inject more sample, seemingly the apparent molar mass increases as well. So what is going on here? This is a classic example of self-interaction, right? At the very edge of the peak, the concentration is lower. Hence, Remicade appear more dissociated. This is the monomer molar mass. At the apex of the peak, the concentration is higher. So there's the concentration induced self-association going on, making the apparent molar mass higher by the apex of the peak. And of course, as you inject more and more, they follow the same trend with higher and higher apparent molar mass. So really adding MALS to SCC workflow can provide early warning for potentially problematic self-interactions. So if you see this, that means that there's some sort of a self-interaction going on and it's dependent on concentration. So they can imagine in the final, say, UFDF or TFF stage, when the concentration goes really high, you might see problems with stability. And simultaneously, the characteristics of the self-interaction could also be part of a stronger evidence of biosimilarity between antibody drugs. And another classic application for adding MALS to SCC is to look at fragment and aggregation. So of course, as we are able to measure molar mass in the native condition, the native fragments and native aggregates can be analyzed with ease. And this is quite different from, like, say, an intact mass spec. The idea is that here we're able to analyze under true native conditions. So basically your final formulation condition could be your mobile phase without any need for special prep of, say, ions or solvent exchange. So now I'm going to switch my topic slightly. Right, this is just naked antibody so far, be it fragment, reversible association, or irreproducible elution time. Now, a lot of antibodies are also conjugated, right? It could be pegylated, it could be glycoproteins, right? And for other proteins, not necessarily antibodies, could be also protein polysaccharide conjugate vaccines, could be member proteins, it could be protein RNA DNA complexes, and also as a derivative antibodies, right? We might be looking at antibody drug conjugate, ADCs. So light scattering also offers a conjugate analysis that is able to look at the molar mass just from the protein, molar mass from the modifier, and the total molar mass. So whereas earlier you see one molar mass line, in the conjugate analysis, you see 3. And from that molar mass, then we're able to calculate the stoichiometry or the composition. So as an example of pegylated proteins, all right, where we're looking at stress samples, stress sample number 1 and stress sample number 2. Yeah. In this case, the stress sample, that's stress sample number 2, had more this dimerization event, right? So the quantity of this dimer is higher. However, the stoichiometry of the protein to the pegylation held, right? So here is a 40 kD protein that become 80 kD protein dimerization. At the same time, this 20 kD pegylation also doubled. So the stress didn't change the stoichiometry, i.e., say the pegylation fallout or hyperpegylated, is a simple dimerization. Now, we can also expect in certain other cases, say you not only have a dimerization or aggregation event, your conjugation might also, maybe thermally unstable and get chopped off, right? All of these can be captured in a single assay. Pegylation or glycosylation in this case, glycosylation could also be different between how you produce them, right? Produced from mammalian cell or produced in the insect cell. So in this case, from two different kind of cell lines, the protein molar mass stay constant, right? So we have produced the correct protein amino acid sequence. However, the glycan profiles are very different. And so again, this is another good way to add additional dimension of analysis by adding MALS to SCC workflow. Now, moving on from pegylation or glycosylation, I think a lot of you probably are working on some sort of map derivatives. So I took liberty to also look at antibody drug conjugates. Following the same logics, right? We're able to look at the molar mass from the map, and we're also able to look at molar mass of the drug linker, which then we're able to calculate the DAR ratio. Now, for Acitrus as an example, this is a cysteine conjugated ADC with a nominal DAR value of 4.2 plus or minus. And if we're running on SCC mode, so that's science exclusion chromatography with MALS, right? We see the main peak, we see the dimer, and the high molecular weight. And we know it's dimer because the molar mass of the protein exactly matches our expectation of a dimer. And then we also see the drug molar mass, or drug linker to be precise, and from which we can calculate the DAR of every peak, right? We can calculate DAR of the mean species. We can also calculate DAR of the dimer and the high molecular weight. And something quite interesting is happening here. The DAR value overall, as measured by secmoles, is 4.1 plus minus 0.1, with the COA to be 4.2. But if we're just looking at the dimer and its DAR, we see that it's 5.7, and it make up 2.4% of the total mass. And the high molecular weight aggregate has an even higher DAR at 7.2, and that make up 9% of the total mass. This tells us this Acitrus drug substance has a propensity for aggregation for high DAR species, which is logical or rational, as in the high DAR species are more hydrophobic and they tend to aggregate more, which are reflected here with a higher average DAR value. Now, if we are to take the same Acitrus DS and running in HIC mode, so that's hydrophobic interaction chromatography with multi-angle light scattering, then we're able to better resolve the different various DAR species. Whereas here is resolution by size, here is resolution by hydrophobicity, and the higher DAR species eludes later. Similar story here, we're able to look at the protein molar mass in blue and the drug molar mass in red. And from the ratio, we can easily identify what they are. This is DAR2, DAR4, DAR6. And also we have some free map right here and a map fragment. Both contain no drug at all. DAR8, unfortunately, is also co-eluting with a whole bunch of aggregates. So you can see the molar mass start to shoot up. And this is in logarithmic scale. So they shot up quite a bit. And we have identified trace amounts, a small amount of DAR5 and DAR3 species, most likely due to some sort of deconjugation event from DAR4 to DAR3 and DAR6 to DAR5. And this is a good way in process development to try to optimize process to eventually the goal to eliminate these side species. So secmoles enables a routine DAR and aggregation analysis for in-process samples on the native condition, whereas HICMOLs add another dimension to look at in-process samples. For a lysine conjugated ADC, and then we use a Catzilla drug substance. This Catzilla DS, of course, we see the main ADC species right here, and a dimer. And this average DAR of the entire species and the DAR dimer is about the same, which tells us, unlike earlier example, citrus, this Catsilla DS has no propensity of aggregation for the higher DAR species, right? The dimer DAR and the mean DAR, dimer and the mean species has the same DAR value. And in reverse phase chromatography modes, we're still observing the same trend. Now, of course, the dimer melts away and become just monomer species. So again, the SECMOS enables a routine DAR and aggregate analysis for in-process samples and for quality control, whereas you can add additional mode, try to see the different insight. For instance, here we are seeing a slight uptick in drug molar mass in SEC mode, by the way, whereas the protein molar mass stay flat, meaning that this is a pure monomer, yet there are some subtle heterogeneities in the drug linker. However, because SEC fundamentally separate based on size, RH, the differences of a few more drug linker does not change the size much, and hence the separation here is already poor. Now, in reverse phase, we should be separating based on the hydrophobicity, and we are still seeing this upward trend. So that signifies indeed this is a low DAR species and high DAR species. However, we might have to optimize the reverse phase gradient a bit more to see if we can pull out a bit better. Now they're still sort of co-eluting from low DAR to high DAR. So that is a matter of future RPC optimization. Now, lastly, we can use SECMOS to provide bridging data that translate, say, a DAR analysis or aggregation analysis from mass spectrometry to a more QC-friendly, such as SEC MALS. So here, as we can see, in 50 millimolar ammonium acetate in red, that is the most common intact mass condition. Whereas here in blue is the Dobecco's PBS mobile phase, so something more native. While we measure the same mean species, this dimer, Catsilla DS, is only present in Dobecco's PBS, signifying this is a constituent native dimer. But if you change it to something else, it might go away. So if you are solely rely on MS to tell you the intact mass and DAR, you might miss this constituent dimer that'll be present in your final product when it's formulated in something like a PBS. So this is a good way to do bridging data that go from R&D, where you have, say, high-powered, high-res mass spec to give you peptide mapping, to give you DAR species profile, to something more QC-ready, such as routine SEC or routine SEC MALS. And as a perk, our software can also give you the extension coefficient of the complex. So with a single assay, we're also telling you the complex UV extension coefficient. So it's not just the VADC or the drug, which both are UV absorbent, but when it's complex form, what's the UV extension coefficient? Which then you can use the UV extension coefficient for such as a UV vis quantitation for concentration analysis. Another kind of conjugate, again, starting from antibody, you can conjugate to drug, you can also conjugate to oligonucleotides. So that's called AOC. In this case, before we have analyzed the AOC, first the customer asking us to look at the siRNA by itself. So in orange, this is the siRNA by itself. We see a pure monomer and nothing else. However, when it's functionalized with the MCC linker, we see this dimer formation. We're not quite sure what's the implication of that yet. This is still a research sample, but nevertheless, just like the antibody case, we can use SECMOS in a regular routine way to analyze the aggregates, or if there's any fragment, as a quality control step. Moving on, this particular customer wanted to synthesize a DAR of 2, or in this case, OR of 2, like oligo antibody ratio of 2 species. And indeed, in SCC moles, we can see a perfect protein molar mass monomer. And we see the protein molar mass of a dimer and protein molar mass of something that's much higher. There's a higher molar mass aggregate. And if we look at the siRNA molar mass, we can see here indeed is the OAR of 2. But buried underneath this tiny little bump, this tiny little shoulder, we see the molar mass of the siRNA drop to 1 copy. So we already know that this is OAR of 2, and this is OAR of 1 co-elution. Just like in the ADC case, SCC separates by size, and small modifications on antibodies, such as some more drug or some more siRNA, does not change the size much. Hence, in SCC, it shows up as a co-eluting shoulder peak. And the identity of this bump is identified as OAR2 dimer because we see 2 copy of a antibody molar mass and 4 copy of the siRNA molar mass. So we know this dimer is also holding up the stoichiometry of OAR2. And actually, interesting enough, even the high molar weight, high molecular weight aggregates still holds up the stoichiometry of OAR2. So this chemistry is very selective in making OAR2. Even the aggregate has the consistent stoichiometry. However, if we move on to using ion exchange, so in this case, we're separating by charge, right? And the siRNA being highly charged now enabled the ion exchange to pull it apart. So now we can see a fully baseline resolved OAR2 1 species right here, right? This correlates to this part. And now, because we're separated by charge, the aggregates do not separate very well anymore. And so OAR2 and a bunch of aggregates start to co-elute. Nevertheless, combining sec moles and AEX moles provides a comprehensive quantitation of AOC payload variations. And so the hypotheses were raised. What really causes this OAR1? So we have done a stress study incubating the AOC at 37 degrees Celsius for 24 hours. And we can see that there's increase in the mass fraction of this AOR species after stressing. So again, AEX moles can be used in R&D and PD to screen for stability. And now this same study can be done with even just naked antibody, right? After stress, are you seeing the same peaks? Are the peaks the same molar mass, i.e. the same identity? And is there any changes in quantity, that is mass fraction? And all of this can be done in a single assay. And lastly, and this will be the last set of examples I brought today, is GLP-1s. So glucagon-like peptides 1 analogs. Similar to antibodies, at the end of the day, in the eyes of moles, multi-angle light scattering, proteins and peptides are the same thing. They're all made by chaining a bunch of amino acids together. So the same analysis can be done for peptides. And here we have liraglutide drug substance and the semaglutide drug substance. And under the native formulation, we can quickly identify that liraglutide DS, in this particular case, we have a hexamer and a 13mer. And how do we know? We know it by the native molar mass. And since we already know the monomeric molar mass, that quickly tells us the identity of the peaks. So in native condition, liraglutide does not exist as a monomer. We have not seen any monomer on the entire chromatogram. They natively exist as some sort of a higher structure bundles, hexamers, 13mers. And for semaglutide, we also fail to observe any monomers. Again, in native condition, these peptides tend to bundle together. And the way they bundle together is complex. and it requires dedications in analysis, comparing, say, biosimilars to innovators. And the semaglutide in this particular case exists as a trimer-tetramer equilibrium, right? And this is a reversible association. When the concentration is lower on the fringes of the peak, it exists as a trimer, and at the high peak is existing as a tetramer. And we can also look at the hydrodynamic radius as a additional perk. And just like antibody case, the identity, the molar mass identity, I shall say, is independent of elution time. So now that also enables us to screen for formulation. So for instance, for this Wegovy drug product, right, if we are to shift its mobile phase, let's say, i.e. screening for different formulations, right, it shifts the peak, but we are still getting, accurate molar mass measurement. And in this particular case, when we increase the pH from 7.4 to 8.1, and when we drop the molarity of the ions, so dropping the sodium chloride ion from 140 millimolar to 0, we're shifting the trimer-tetramer equilibrium, right? In this native condition, it is shifted towards more tetramers, whereas in this condition, it is shifted to more trimers. Now, similarly, we have tried two different formulations, but the liraglutide drug substance has no changes at all. It still exists as a hexamer bundle and a trimer bundle. Actually, now I'm looking at it, it still exists as a 13mer bundle, but here is more of a hexamer. Here is more like a maybe 7mer, which is quite unique. So mobile phase might impact aggregation profile, and it also changed the retention time. But now with MALS added to SEC, the retention time doesn't really matter. So that will give you the confidence in identifying what kind of aggregation profile that you're shifting your sample as a function of different conditional screenings. Now, a big analysis is always comparing DS with DP. And in this particular case of this drug substance compared to Wegovy, the trimer-tetramer equilibriums are exactly the same, right? So this offers a very strong evidence of biosimilarity in vitro, where even the reversible associations are reproducible. However, in this case, the DSDP are quite different. This drug substance of liraglutide contains a unique 13mer. However, that's absent in this DP, whereas this DP has this very large aggregate, right, tens of millions of Dalton, that is absent in the DS, while they share the same core hexamer mean product. So again, you can use light scattering to very quickly generate high quality data on the API DSDP similarity. And as elucidated earlier, these higher degree aggregates or higher order structures, some of them might be reversible, and some of them might be irreversible. And that's also the same case of, say, the Remicade study, as I have mentioned earlier, for antibodies, right? There might be some reversible, irreversible interactions. So in this particular case, you're still using liraglutide example. Now we see the hexamer, the 13mer, and as we inject more and more, so introducing higher concentration, the molar mass stayed consistent, i.e. this structure is irreversible. However, if we are to look at semaglutide and we are injecting more and more, this trimer-tetramer equilibrium can be shifted, right? As we inject more, there's apparently higher molar mass, which means there's a shift towards more tetramer and vice versa, right? So we can use CEPMOLs in the native condition to look at the reversibility of these higher order structures. And again, this also acts as a stronger evidence yet for biosimilarity. After looking at the reversible or irreversible interactions. Another layer of biosimilarity comes from covalent or non-covalent aggregation. So if we are to denature these peptides under acetonitrile, high organic, and low pH, pH of 4, then we break apart these higher order structures. And then instead, now we're looking at semaglutide DSDP. When they're all denatured, it becomes 100% monomer. Great. So there's no covalent link aggregates. However, for liraglutide, if you look at DSDP, indeed they are. There's 97% monomer, but they are dimers and trimers that withstand the denaturing condition. So they are covalently bound to each other. So overall, when you're comprehensively looking at denaturing segments, we're looking at native segments that give you the best biosimilarity data or profile of the various fragments or aggregate species there is between the DS and DP. So overall, we have examined peptides. We have looked at conjugated systems such as AOCs or ADCs. And we have a look at native maps and how that you can use segments to enable routine testing for quality control, but also in your R&D lab for biosimilarity studies, looking at the identity of these species, whether it's native condition or denaturing conditions. And we have looked at ADCs and AOCs for more complex modalities for stoichiometries. And we have looked at glycosylation pegulations. So these kind of other modalities, if those are of interest to you, can also be analyzed. So all of these parameters, right, can be analyzed in a single run, and we call this multi-attribute quantitation. So in the past, you might be running different assays of different technologies and different instruments. Now, a lot of the assay can be consolidated, simplified in a single workflow. And lastly, now we support the full waters and wired technology integration, and all of these are powered by QC-ready systems and with full 21 CFR Part 11 compliant softwares. So with that, I'm happy to take any questions.
Thank you, Dr. Yo. I think that was a very impressive talk. I really, the case studies that you took, the examples that you took were really interesting. So we do have a couple of queries in the chat box. So let me start. Yeah. So we have a question from Siley. And what I'm asking is that if we do not have a standard for the column calibration and normalization of detectors for proteins or maps, what should be done in that case?
Thank you for the question. So I might break apart this as two questions. One is what happens if you don't have a standard for column calibration? The answer is no problem. In fact, you don't need to have a column calibration at all with MALS. However, if you do choose to use traditional GPC or SEC, then yes, that's an absolute. And you want to go back to the 4 critical assumptions. I might flip back the slide here, just so everyone's looking at the same thing here. You'll want to be very, very careful that your column calibration and your analyte should preferably satisfy all 4 assumptions simultaneously. Now, if there's a particular workflow you want to explore, I'm happy to take some questions, later. You might ask, venture centers, I might be able to send you more information via email. Now, the second question is, now, assuming you're looking at SEC MALS and you don't have a detector normalization standard, that will also be problematic, but it's easier to overcome because there's many ways to normalize detector. One is using something that's just very small. USP has a BSA standard following their new general chapter 23329. I want to say that. I might have to check. Maybe I will update everyone. a new general chapter is coming out from USP that talks about the segments, and then they also have normalization standard that come with it. Then that's universally applicable. It doesn't really matter what kind of analyte you're gonna run, be it antibodies or even peptides, the same and normalization standard can be used. And then we generally will only normalize detector for particular solvent. So if you don't change your mobile phase, then the normalization coefficient will stay. Could be years. You only need to redo it once.
Sure. I think that was very informative because I think
Mm.
there are certain misconceptions that if you are using a MAB, then you need a different, normalization molecule. And if you're using a peptide, then you need, say, a smaller this. But I think you have clarified on that, and that makes sense. So, but would you just like
Uh,
to elaborate on the solvent system? So you said that
mhm,
unless the
mhm.
solvent system is changed, then the normalization is not required. So would you like to elaborate on that part of the information that you
Sure,
just
sure,
shared?
sure, sure. I'd happy to. So yeah, so it's a misconception that say proteins and peptides like you have to have a different normalization. That's not the case. Normalization is tied to solvent, mobile phase. The reason is that different mobile phase composition would have different refractive index, right? Light will always refract. So to accurately calculate the light scattering intensity, we need to know how much the solvent is refracting light. And that is normalization. So in fact, normalization is tied to a solvent, not to an analyte. If your, say, antibody is using PBS and your peptide is also using PBS, great. It's the same normalization. But if it's a very different mobile phase, like say 10% sucrose in one and 20% alcohol in there, yes, that's a different normalization coefficient.
Great, great. I think that was a good clarification. Siley, I hope that is clear. And if you have any follow-up question, maybe you can put it in the chat box while I move to our next question. We have another query, and this is from, oh, okay. I don't know. I think there was a question from Chaitanya, and he was asking if whether we have. I don't see it in the chat box, but I remember reading it. He says that did we? Did y'all do any experiments with virus-like particles for using SEC MALS?
Can you repeat what kind of particles?
Virus-like particles, VLPs.
Oh, VLPs. Yes, yes, it's
Yes.
actually a common usage of it. VLPs such as synthetic polymorphism or natural VLPs, or like say a big one like L2 protein complexes for HPV vaccines. Um, in fact, light scattering for HPV vaccine is regularly used and commercialized. So I don't see any particular problems with VLP analysis. We generally treat it as just another kind of protein, big ones.
Sure. So I'm sure there'll be some white papers or
Yeah,
application notes on this, right? So Chaitanya,
yeah,
probably you can visit the Waters VAT website, and I'm sure you'll find more
yeah,
literature
yeah,
over there.
yeah.
Yeah. Yeah.
Or just Google Scholar search. There's plenty literature references of light scattering and the VLPs.
Sure, sure. Thank you. Any other questions from our participants? We can spend another few minutes. Like maybe if you all have anything, you all can just unmute and ask because we have Dr. Leo with us, and I think he has extensively covered all nice molecules. The very interesting molecules. So I think we should take up could make use of this opportunity to have this interaction with him. So any questions? We can take one or two questions more. Or if that's not the case, I think we can always write to him, not an issue. Maybe I have one query, I think, because I was quite engrossed in the session. Wanted to understand more about DAR calculation. So you had a
Mm.
couple of slides where, SEC is being used for that, and you had a comparison with HRMS as well. So I wanted to understand one, the regulatory requirement, like would they accept both the DAR calculation by HRMS and MALS? And I know the slide said that, with MALS, it was slightly more better, but what is your take on this, or on the comparison on the data that we receive? Because I think TAR calculation is very essential, and many clients have been,
Yeah.
and uh, think this wanted to know your take on this particular application.
Of course, yes. Now, um, star calculation from mass spec, would it be still considered to be the gold standard,
Okay.
right? All the approved, FDA approved, so released in the US, star ADCs, every single one of them had DAR from mass spec.
Okay.
And there's only what 15, 16 of them. So um, regulators are accustomed to see that. And of course, Waters Corporation as an entity, we also make awesome mass spec. So uh, the problem with that, or the limiting case, is mass spec for DAR analysis are usually done with reverse phase, right? And
Okay.
under
Right.
reverse phase conditions or some sort of compatible solvent conditions, and then you look at the DAR distributions. Now, SECMOS for DAR analysis is very recent. We have only commercialized the process or the analysis beginning of the year. So I don't think any regulators have seen
Okay.
formal IND filings with SECMO DAR. We are
Mm-hmm.
working very closely with both FDA and USP on this. So the value
Mm-hmm.
set is not exactly in quality control release assay,
Okay.
but rather as in process development, those in-process samples where you need to get R of plus or minus 5% accuracy towards NMS in a more rapid response way, where you are generating a lot of process samples and they are all in various native and or high ions or process conditions. So we have asked customers that it is incredibly time consuming to then have a offsite or on the assisted site have a mass spec to analyze process
Mm-hmm.
samples. The volume is really large.
Absolutely.
Sample prep
Mm-hmm.
is really difficult. So you want
Mm-hmm.
to bring most analysis in a routine deployment to look at these process samples. Of course, in R&D, we'll have high-res MS, and in QC, probably will still have batch-based
Mm-hmm.
higher. You probably will not have a high-res MS on every single
Right.
run, but batch by batch, you will probably will do lot testing because that's how the regulators has been accustomed to see the release data.
Right. So I think that was an interesting,
Mhm.
take on the DAR calculation. Definitely in process development, I think this will come in very handy. And
Mhm.
just one follow-up question. So this is the software readily available for this, or like does it come with the
Yes.
existing module itself?
Yes, the software is so the DAR part of the analysis in Astra is commercialized January this year.
Okay, so 2025. Sure,
Yeah,
sure. Good to know that. Yeah, I think that's there. Um, if you permit, we have one more question, or maybe we can take that offline. I think I see it in the chat box right now. So would that be okay?
sure.
Yeah. Yeah. So again, it's from Saili, and she's saying that normalization of detector with 3 nanometer to 3.9 nanometer will be helpful if we do not get the standard. Does this impact the molecular weight calculation drastically?
Again,
So,
that's a very good question. Goes into technical details of how, like, what is normalization. So, yes, if you have anything that is like 3 nanometers, that works. The idea is that for something significantly smaller than 10 nanometers, they are said to be isotropic scatterers, meaning the light that scatter from it would be equal in every direction. And that is very important. That is the basis of the normalization procedure. It has to scatter light equally every direction. And anything that is significantly smaller than 10 nanometers is isotropic scatterer. So if you can find anything that is 3 nanometers or even smaller that act as a normalization standard, if you run out of, let's say, BSA, lysozyme, insulin, the common few, dextrans, you grab something else that is less than 10, like very smaller than 10, like polyethylene oxide even, yes, you can normalize on it.
great, great. So I think, yeah, Saili, I think that clears the the doubts that you may have. And I think with that, we now come to the end of the session. I take this opportunity to thank you, Dr. Leo, for this very, very informative session. Thank you for accepting our invitation and delivering on this session. And as you mentioned, like this topic was very vast, and hopefully we'll be able to collaborate on more sessions
Sure,
in the
sure.
future. And
Sure.
participants, thank you as well for participating. I have shared the feedback link. Please do give us your valuable feedback so that we can incorporate all the suggestions in the upcoming talks. So thank you all of you all once again, and see you all in the next session, which will be next month with a new topic. So thanks a lot again for your
All
participation.
right. Thank you. Thank you for Venturesome inviting me. Thank you, Anna, for hosting
Yeah,
me. My pleasure to be here. Take care,
yeah.
everybody.
Thank you. Have a good day ahead. Yeah. I'll request all participants who are still there to kindly give us your feedback. I'll really appreciate it. It will help us to, improvise on the